Everyone. All right. Good morning, everyone. My name is Murali Temburni, and I'm an associate professor at Delaware State University. Previously, I was a postdoc at the University of Delaware, so I have been associated with UD and with DSU for a long time now. And I'm happy to be an IMBRI pilot PI and thankful for the support. So as my title suggests, my lab is interested in understanding the interactions between astrocytes and neurons, and we are trying to understand how astrocytes influence neuronal networks. Yes, laser pointer. Right. So, as we're all neuroscientists, you understand that neural activity begins very early during development, and initially this activity is random. This is an example of how random activity looks like in an in vitro multi-electrode array recording, the examples of which I'll show you in a few minutes. So this is what random activity looks like. So initially the activity is random, and over a period of time in vitro cultures, this activity becomes highly synchronized. So these are just 4 of the 120 electrodes that we use for recordings. So what is the meaning of this activity? We really do not know yet, but random activity mostly occurs in the awake brain when the brain is processing sensory information. And synchronous activity happens mostly seen during sleep and anesthesia in the adult brain, but it's thought to be necessary for the functional organization of the brain. The brain is functionally modular. And also, we see this kind of synchronous activity in the awake brain during pathological conditions like epilepsy. Currently, the focus in the field has been on neurons, as we all know, but it was around 20 years ago, more than 20 years ago, that the idea that astrocytes could modulate neurotransmission took root. And this was a PNAS commentary that we wrote when the late Ben Barres published the paper showing astrocytes being involved in neurotransmission. So this idea, and ever since I was interested in understanding how glia, particularly astrocytes and neurons interact. So astrocytes, to tell you briefly, can sense, these are synaptic astrocytes, they can sense neurotransmitter that's released at the synapse, and they can respond by elevating calcium. Astrocytes do not contain ionotropic channels. They only contain metabotropic channels. They only express metabotropic channels. And the increased calcium flux in the astrocytes can cause them to release their own chemical transmitters. These are called gliotransmitters. Glutamate, ATP, D-serine are a few that have been identified. So coming to my system, we are interested in understanding the interactions, and we use the chicken embryo and the chicken forebrain and the optic tectum as our model, because these are very easy to culture and they're much cheaper than using mouse. So here are our cultures. So we have these 3 conditions where we grow neurons and astrocytes. Control, in which both astrocytes as well as neurons are present. And then we culture pure neurons by killing off the astrocytes using a mitotic inhibitor, 5-fluorodeoxyuridine. Astrocytes are mitotically active, so they are killed, but neurons are post-mitotic and they survive. They are not affected by FUDR. But however, to keep the neurons alive for longer times, we add an astrocyte conditioned medium, we call it ACM, and this contains all the growth factors that are necessary to keep the neurons alive for long term. So our current conditions are FUDR treated. The treated is FUDR treated in the presence of ACM, and control cultures only in the presence of ACM, because the cardinal rule in experimental biology is that you can change only one thing at a time. So these are our control cultures and these are our treated cultures. So we grow these cultures on multi-electrode arrays. So we currently have the multi-channel systems containing 120 electrodes, but this was the older version, the MET64 system, which has 64 electrodes. So now we can record activity, and I'll show you how this activity looks like. So these are control cultures at about 2 weeks, 14 days in vitro. And you can see that the activity in all the 64 channels is mostly random. I hope you guys can see the spikes. So these are 50 millivolts. on the y-axis and about 200 milliseconds on the x-axis each division. But by these control cultures, by 21 days, by 3 weeks, they now become fully synchronized. And however, uh, FUDR treated cultures, that is those cultures that lack astrocytes, even at 21 days, and then we culture them beyond 3 weeks for up to a couple of months, they never synchronize. They continue to show random activity. So indicating that astrocytes are necessary for the neurons to develop synchrony. Uh, we did, I'm not showing the data, but we did patch clamp electrophysiology and showed that these FUDR treated cultures are indistinguishable from control ACM cultures in all their electrical properties. And we also, we also actually added back astrocytes to these cultures and then saw a bit of synchrony return. However, these are difficult experiments to do, and we couldn't repeat them many times. So in the absence, but to summarize, in the absence of astrocytes, as long as they're supplemented with the conditioned medium, neurons are healthy, but they fail to synchronize. So we conclude that astrocytes are needed for generation of synchrony as well as the maintenance of synchrony, because when we remove astrocytes at different time points, synchrony is lost. In a couple of days of removing astrocytes, the neurons all start firing randomly again. So how does this happen? So our hypothesis is that the GPCR coupled receptors on astrocytes via calcium elevations control the development of synchronous activity via exocytotic release of neurotransmitters. So by now, astrocytes have been shown to express the metabotropic glutamate receptor, the P2Y1, the purinergic receptor, and the GABA B. All of these are metabotropic. They're not ionotropic channels. They're metabotropic receptors. And we decided to use in order to test their role in the control of synchrony or network activity, we decided to use a dominant negative strategy, use a molecular approach rather than a pharmacological approach. So our first one was we targeted the metabotropic glutamate receptor. And then our idea is once we have these line of astrocytes, we can co-culture them, put them back and co-culture them with neurons and MEAs and then look at network activity. So these are the standard pathways that have been identified and characterized in astrocytes. So we can also use pharmacological agents to activate or block them. So I'm going to show you data for the mGluR now. So our model is something like this. So activity that is detected at a synapse by an astrocytic process via the mGluR receptors will cause an local elevation of calcium, and this calcium elevation, once it crosses a threshold, will travel down the process to the cell body and then cause a calcium-induced calcium release. Now this large amount of calcium that's released from intracellular stores can do two things. One, it can get into other astrocytes via astrocytic gap junctions and then activate other astrocytes in the network, or it can cause the release of gliotransmitter like glutamate or ATP or GABA or D-serine. So this is the model that we wanted to test. So in order to do that, first we targeted the metabotropic glutamate receptor. I'll not go into the details, but we expressed a truncated version of the mGluR in these astrocytes, and then we look for calcium activity using an engineered protein called GCAM. These are the rage now. So in the past we used to use chemical detectors of calcium, but now GCAMs are the way to go. So it's a very fast calcium detector. So it can detect calcium flux on the scale of milliseconds. So here we can see that these are controlled astrocytes just expressing the GCAM6F. The addition of glutamate will cause elevation of calcium easily. Just I'll wait for the movie to stop. Okay. In the second panel here, you see the M-blue or dominant negative being expressed along with GCAM6F, and there is clearly a much diminished calcium elevation in these cells. Towards the end, we wanted to make sure that actually that these cells are capable of, they do contain calcium stores. So we add ionomycin, which is a calcium ionophore, which causes the large calcium release that you see in the end. So we repeated this experiment multiple times, and so we can clearly conclude that our dominant negative is working. The next thing that we wanted to check was glutamate release by astrocytes. So Toby, a graduate student in my lab, started this project. So astrocytes have been shown to express the snare complex. They were shown to have vesicles. We call them exocytotic vesicles containing gliotransmitter. They also have the snare complex proteins. So one of the way is when calcium enters the neuron. This is what happens in neurons. When calcium enters the neuron, causes the conformational change in synaptobrevian contact syntax in the snare complex, and then causes the fusion of the vesicle, and neurotransmitter is released. So Toby decided to use the same strategy, a dominant negative strategy, and we basically express the truncated version of synaptobrevian, only the cytoplasmic domain. The membrane spanning, the vesicle membrane spanning domain is deleted in this. So he expressed this. So what we expect is that synaptobrevian will bind to syntaxin on its own and then prevent the binding of the endogenous synaptobrevian with the snare complex. So we sense glutamate by using this extracellular glutamate sensor called the glue sniffer, and we got this from Dr. Lauren Luger's lab, and it works beautifully, as you can see. So these are controlled astrocytes, and when we increase calcium in these cells by using ionomycin, we can see that that's when ionomycin is added. Glutamate is released on the surface, and we see it all over. But ionomycin kills the cells, is a poison, is an ionophore, but towards the end, in about a minute or so, the astrocytes start dying out. VAMP2 dominant negative expressing astrocytes have much diminished calcium glutamate release. That's when ionomycin is added. And the astrocytes dying after the experiment. So Toby repeated this experiment more than a dozen times, and we can clearly see that both our dominant negatives, the M-GLUO dominant negative and the VAMP2 dominant negative, are working. So this is where we are right now. Now the next step is to grow these astrocytes and to culture them and then add them back into neurons. For the INVIT project, we now decided to target the other pathways, the P2Y1 pathway as well. So we are going to be targeting not the receptor itself, but a scaffolding protein called NHERF2, which is necessary for downstream signaling. So the scaffolding protein NHERF2 is necessary for PLC beta 1 downstream signaling via the P2Y1 receptors. So a dominant negative that lacks the PDZ domain, first PDZ domain, acts as a truncated version of NHERF2 acts as a dominant negative. So we are in the process of making this. We're also targeting the GABA receptor pathway. So GABA receptors, we are using a slightly different strategy here. We're going to make point mutations in the GABA B2 subunit. And then we want to express them using lentiviral vectors, just as we did the M-GLUO or dominant negative. So ultimately, what is our goal? Once we get these dominant negatives, we would like to culture neurons and then add back the dominant negative astrocytes with these neurons, neuronal progenitors dissociated from the chicken optic tegment forebrain, and then culture them on on multi-electrode arrays and then look at network activity. I haven't talked about this right now, but we also have developed an optogenetic tool called melanopsin. We expressed melanopsin and also showed that when you turn on blue light, we can actually cause calcium elevation in these cells. So we can also do optogenetics on these using these cultures. So we just bought the instruments in our electrophysiology core, and I'm really excited to try this in the summer. We also can actually want to take these astrocytes and inject them directly into the optic tectum. So I learned this technique in the lab of Dr. Denny Galileo when I was a postdoc, where he used to inject human glioma cells. He still does into optic tectum, and they can get into the network and they can spread. So others have shown that you can inject fully matured astrocytes into developing brains, and they will actually integrate into the network. So we're going to try that this summer. So I'm going to stop here, and I would like to acknowledge my collaborator, Dr. Zakfasu, Dr. Harrington, and my lab members. And then I'd like to acknowledge the Cobri Pilot Grant. I'm sorry, I forgot to mention the MRDA. I'm also thankful for the MRDA grant, which gave me one course time release last year and the new MD pilot. I'll stop here, and then if you have any questions, I'll be happy to answer them.